My name is Caitlin, and I am a research associate within the core services located in the Department of Chemistry at the University of Alberta. Today we will be talking about the characterization of carbohydrates using mass spectrometry. Carbohydrates are key for many biological processes. They interact with lipids, proteins, antibodies, and they participate in cell signaling. Unlike nucleic acids or proteins, the biosynthesis of carbohydrates is not template-driven, which means carbohydrates exist in nature as a heterogeneous mixture that is highly complex. In addition, there is no way to amplify a carbohydrate of interest in the same way that it is possible to amplify proteins through overexpression or amplify nucleic acids through the polymerase chain reaction. Therefore, to analyze naturally occurring carbohydrates, researchers must often deal with small quantities of heterogeneous material that is isolated from the natural source. Due to the high sensitivity and compatibility of mass spectrometry to handle heterogeneous mixtures, it is an essential tool to analyze carbohydrates. Today, we will be talking about the compositional analysis of carbohydrates, as well as the structural analysis of carbohydrates. And finally, we will talk about coupling mass spectrometry techniques with various separation techniques. In compositional analysis, the primary information you receive about your carbohydrate is the composition or the chemical formula of your sample. This is useful for the confirmation of identity or to check the purity of known samples, but it does not provide you with any information about the fine structural details that you need to fully characterize a carbohydrate sample. Fast atom bombardment, or FAB, is a gentle ionization method which keeps the oligosaccharide of interest intact. This ionization method works well for acidic oligosaccharides in the negative mode, particularly those that contain sialo or sulfate groups. However, it suffers from low ionization efficiency and the ion signal is typically weak. Additionally, the detectable oligosaccharides are those of low molecular weight. The way that FAB works is that the sample is mixed with a non-volatile liquid matrix that allows for a consistent ion current to be sustained. It reduces the damage made to the sample by absorbing the impact of the primary beam and keeps the sample molecules from aggregating. Glycerol is a common liquid matrix that is used. Once the sample is mixed with the matrix, the sample is introduced into the FAB analysis through an insertion probe. A beam of atoms, typically xenon or argon, bombard the sample matrix. The exact mechanism of ionization is unknown, though it is proposed to be similar to that of MALDI. Today, FAB has largely been replaced by ESI and MALDI. Matrix-assisted laser desorption ionization imparts very little energy to your sample, which prevents extensive fragmentation of the oligosaccharide. Carbohydrate samples can be analyzed in the positive or negative mode depending on the number of acidic or basic groups present on your sample. In addition, the ionization efficiency remains constant regardless of the size of oligosaccharide. However, the ionization efficiency is lower in MALDI compared to other ionization methods, and some oligosaccharides have modifications that have been found to be thermally labile and small fragmentations may occur during analysis. To prepare your sample for a MALDI analysis, a matrix is needed. The matrix serves two purposes. It absorbs some of the laser's irradiation and it helps ionize your sample so that it can be detected by the detector. There are many small molecules that have been well studied as matrices, and a list can be found in the reference below. The first layer of the matrix is spotted onto the sample plate and allowed to dry, forming a smooth, uniform surface. Once the first layer is formed, a small amount of your sample is added to a solution of matrix for layer number 2. This mixture is spotted directly on top of layer 1 and allowed to evaporate. Once the samples have evaporated, the sample plate can be inserted into the machine for analysis. The sample spots are irradiated using an ultraviolet laser. The matrix absorbs the energy from the laser and transfers a small amount of energy to your analyte, which causes your analyte to be ionized. There are some disadvantages to MALDI in the analysis of carbohydrates. Specifically, carbohydrates typically have a low ionization efficiency in MALDI, meaning that this method of detection can suffer from low sensitivity. Some sugars, sialo sugars in particular, are thermally labile and may degrade during analysis. The mass obtained from a MALDI analysis alone is not enough to identify your carbohydrate. You must use this method in tandem with other identification methods to confirm identity. Electrospray ionization, or ESI, is a gentle method of ionization that keeps oligosaccharides intact. This type of ionization couples well with separation methods like liquid chromatography or ion mobility. However, the sensitivity of ESI decreases as the glycan mass increases. To produce ions of your carbohydrate of interest, a very dilute solution of your sample, around 1 to 10 micromolar, is injected through a thin diameter needle placed near the inlet capillary of the mass spec instrument. A potential is applied, and the difference in potential between the tip of the needle and the capillary is coupled with very high temperatures that generates finely charged droplets. An inert gas is used to vaporize the solvent as they are aspirated into the mass spectrometer. Generally, positive and negative ions are produced in this method, and multiple charging may be observed. Neutral species have very poor ionization efficiencies in this method. To elucidate structural details of carbohydrates, tandem mass spectrometry based on collisional-induced dissociation must be used. This is a series of low-energy collisions that are used to break apart the glycosidic bonds. The primary sites of breakage are on either side of the anomeric hydroxy group. In the example above, the trisaccharide can be broken into the B1 and Y2 ions, or the C1 and Z2 ions. This type of bond breakage can occur at any anomeric site, resulting in the B2Y1 ions or the C2Z1 ions, or even the B3Y0 and the C3Z0 ions. Higher energy collisions can result in cross-ring fragmentation, which result in the A and X ions. Each of these ions can be analyzed to provide information about the sequence of the monosaccharide linkages and locations of potential modifications. The monosaccharide stereochemistry and anomeric conformation cannot be elucidated using MSMS alone. To elucidate these structural features of oligosaccharides, higher stages of tandem mass spec are needed in conjunction with the use of standards and databases. The de novo structural determination of carbohydrates involves breaking the oligosaccharide into overlapping segments of disaccharides. This process is done through successive rounds of collisional-induced dissociation. In this process, the sample is injected into the mass spectrometer to produce a parent ion. That parent ion then undergoes in situ fragmentation to further break down the oligosaccharide into disaccharide fragments. Each disaccharide has a fragmentation fingerprint, which can be compared to known disaccharide fingerprints found in databases. There are many databases online that contain fragmentation patterns of standard disaccharides. One such database is glycosciences.de. By comparing the disaccharide fragmentation pattern with standard fragmentation patterns, the individual disaccharides can be identified. In reality, this is a very complex process. In addition to the Y and C ions shown here, the Z and B ions are also generated. In addition, A and X ions are produced by the dehydration or cross-ring dissociation, and they must also be considered in order to determine whether or not your sample is linear or branched. By further adding to the complexity of structure determination is the presence of many different types of linkages. Specific linkages such as 1,4 and 1,6 produce different fragmentation patterns than a 1,3 linkage. A detailed description of the different types of fragmentation that can be observed and the process of de novo carbohydrate structure analysis can be found in the reference given at the bottom of this slide. Let's take a look at an example of using higher order tandem mass spectrometry for the de novo structural analysis of carbohydrates. In this example, we will be talking about the trisaccharide Panose. The first step is to obtain a mass spec of your parent compound. In this case, the sodiated parent ion has a mass-to-charge ratio of 527. Based on the fragmentation fingerprint of the parent compound in comparison to databases of fragmentation patterns, it is determined that this trisaccharide has a 1,4 and or a 1,6 linkage. The structures of potential 1,4 linked or 1,4-1,6 linked trisaccharides are shown on the right, and a simplified version of these structures are shown on the left, in which each monosaccharide unit is depicted by a circle. The parent ion then undergoes a second round of collisional-induced dissociation, or CID, which results in sodiated ions observed at 509, 467, and 365. Potential structures are predicted based on the ions observed and are shown on the left. They are a combination of dehydrations and cross-ring fragmentations. Each of these fragments then undergo an additional round of CID, which produces new ions in which structures can be predicted for. This process continues until all the structures predicted based on the ions observed are disaccharides. The fragmentation fingerprints, or the mass spectrum of each disaccharide unit, is then compared to online databases that contain fragmentation fingerprints for all possible combination of disaccharides standards. By matching the fragmentation fingerprints observed to the online databases, the individual disaccharides are identified. The overlapping segments of disaccharides are then pieced back together to determine the identity of the parent compound. Permethylation and peracetylation increases the hydrophobicity of glycans, which increases their ionization efficiencies. Upon acid hydrolysis to the monosaccharide components, the mixture of components can undergo mass spec analysis, which allows for linkage analysis. As most unmodified monosaccharides have the same molecular weight and chemical formula, to determine the linkages, the mass spectrometry analysis is typically coupled with separation techniques like liquid chromatography and the use of chemically synthesized standards to determine the exact identity of the oligosaccharide. If we start to think about the number of different monosaccharides coupled with the number of different types of glycosidic bond linkages possible, as well as the conformation of the glycosidic bond, it is easy to see that there is a large number of oligosaccharides possible for a single molecular weight. In comparison to other biological samples like peptides and nucleotides, carbohydrates have so many structural isomers that tandem mass spec might not be enough to resolve the complete structure of complex oligosaccharides. Coupling tandem mass spec to separation techniques like liquid chromatography or ion mobility allows for separation of mixtures prior to analysis of fragmentation fingerprints. Typically, the separation and mass spectrometry techniques require the use of standards to help identify your sample components. Liquid chromatography couples easily to mass spectrometry. The sample is first run through an HPLC, and each peak that appears on the chromatogram is directly injected into a mass spectrometer. The output is a two-dimensional spectrum where you first look at the chromatogram and then look at the mass spectrum that is linked to a specific chromatogram peak. This method simplifies the mass spectrum of samples that are mixtures of components. One main drawback of this technique is that the column used in the separation portion is typically nonpolar and unfunctionalized carbohydrates or hydrophilic, which results in poor retention and separation of unmodified oligosaccharides. In ion mobility mass spectrometry, carbohydrate ions are separated not only by mass and charge, but also based on their size and shape. The size and shape of the ions impacts the time it takes for ions to drift through a weak electric field that is filled with an inert gas such as helium or nitrogen. As ions drift through this column of inert gas, they undergo collisions with the gas. Compact ions will undergo less collisions than ions with extended structures, allowing ions with the same mass to be separated from one another. The time it takes for the ions to drift through the inert gas is called the drift time, and this drift time can be converted to an instrument-independent value called the collisional cross-section, or CCS. When the CCS is used in conjunction with the fragmentation fingerprints of disaccharides, fine structural details can be determined, such as linkage sites, anomeric conformation, and stereochemistry of monosaccharides. The main drawback of all of these mass spectrometry techniques is that you still require chemically synthesized standards or access to databases that contain the CCS and fragmentation patterns for large libraries of disaccharides. In this example, researchers wanted to identify the ligand that was released by the protein and whether or not it was penta-E or penta-F. By comparing the ion mobility of each ligand separately and then comparing it to the ion mobility of the undetermined ligand that was released, we see that the drift time of the released ligand matches that of penta-F and indicates that the ligand released by the protein was penta-F rather than penta-E. This example illustrates how carbohydrates with the same mass can be separated from one another based on the relative size of their corresponding ions. Today we've talked about the role that mass spectrometry plays in the characterization of carbohydrate samples. We've seen that we can have different levels of detail from different types of mass spectrometry experiments. Compositional analysis gives us information on the chemical formula and molecular weight of a sample. However, it does not provide detailed structural information. Structural information can be obtained from tandem mass spec experiments or by permethylating and hydrolyzing your carbohydrate samples prior to mass spec analysis. Finally, we've seen that we can use separation techniques coupled with mass spectrometry to help elucidate the structure by separating your sample into components prior to MS analysis and thereby simplifying the fingerprint fragmentation spectra needed to identify components within your sample.